Hey guys, have you heard of the term GMOs? I'm sure you have, but do you actually know what it means? In 2018, the first gene-edited crop was released to the markets, and in 2019, dozens more will follow. Now if you're like me, you're about to say, wait a minute, GMOs have been on the shelves for years, what are you talking about? Which is definitely true, but this is an entirely new type of GMO. For centuries, we have been modifying plants and animals through a process called selective domestication. Domestication is basically the same as selective breeding. For example, the pit bull was bred to be a protector. We would take the strongest and the most loyalist dogs, breed them, and then out of those offsprings, we would then select the strongest and the most loyalist, breed it again, and that cycle would continue until we now have what we call the pit bull. Pigs were once wild boars. Cows were once this giant animal called the auroch that kind of looks like a giant bison, and basically every other plant and animal was once domesticated. So this type of modification went on for quite some time until the 1850s when Gregor Mendel came onto the scene. Do you guys remember the Punnett Square that we learned about back in high school? Well, that was thanks to Gregor Mendel. He was most famous for his pea plant experiments, which eventually led to the rules of heredity. He focused on hybridization, which was a more precise way to selectively breed plants. Mendel passed away in 1884, but he wasn't the only one hybridizing plants. In 1898, Garton's Agriculture Plant Breeders was established in England. This was the first company to commercialize hybridized plants and seeds. From that point on, hybridization was the latest and greatest technology up until 1969 when Peter Le Bon introduced his idea of recombinant DNA, which we now call transgenic modification. Recombinant DNA is where you take a DNA strand and you cut out a certain part of it and replace it with another organism's DNA. This then changes the plant to have certain desirable traits. The first time this was accomplished was back in 1973, so it's definitely been used for some time now. In fact, a main thing that we use it for today is insulin production. To do this, they take the human insulin gene, insert it into bacteria, and then that bacteria now creates insulin, which we then pack and sell to people with diabetes. So in 1987, the first description of what we now call CRISPR was developed. CRISPR-Cas9 was then created and hailed the biggest scientific breakthrough back in 2015. Since then, companies have been hard at work trying to be the first to create plants, babies, and animals that are gene edited. Let's say you have the DNA for color blindness. With CRISPR, you can go into the baby's DNA and change the DNA so that way it has great vision, it's taller, or even has things like blue eyes. China has released the first gene-edited babies that are said to be immune to cancer. Scientists have also created apples and mushrooms that don't brown, they've created cows that don't grow horns, and much, much more. In 2018, soybeans edited by CRISPR hit the shelves, and in 2019, much more will follow. So back in 2016, the National Bioengineered Food Disclosure Standard was signed into law. This law says that all bioengineered food has to have a label, or it has to have a scannable link that says View Disclosure. That sounds pretty good, but wait. Upon further review of this new law, I discovered that food only needs to be labeled bioengineered if the main ingredient is actually bioengineered, or if the first ingredient is broth and the second main ingredient is bioengineered. This means that companies don't have to label their food as long as the first two ingredients aren't bioengineered. So some big companies like Coca-Cola, Kellogg's, and Campbell's say they're going to voluntarily put the bioengineered label on any of their products that even contain a small ingredient that was bioengineered. Now this I can get on board with. But again, wait, because there is one more giant flaw in this law. This disclosure standard only applies to transgenic foods, or food that was altered using recombinant DNA technology. CRISPR-edited foods will be exempt from the label, and the USDA even says it doesn't have to go through years of rigorous testing just because it doesn't use foreign DNA. We do know that changing an animal or plant's DNA structure will permanently change the genome for future generations. We know that herbicide-resistant plants have created super weeds and super bugs in the past that require even more toxic chemicals, and we also know that changing the ecosystem usually doesn't go as planned. But obviously, they don't really care because they don't require testing. So what do you guys think? Should CRISPR-edited foods be tracked and monitored? Should we be concerned with our safety since there is no testing? Or should we just go with the flow and see what happens? Either way, please leave me a comment down below just letting me know what you guys think. Like and subscribe if you want to see some more in the future. And guys, have a great new year. Happy 2019. Peace.